Exchange of gases in detail. The exchange of gases refers to the process by which oxygen and carbon dioxide are exchanged between the lungs and the blood and between the blood and the body's tissues. This exchange is essential for the respiratory system to fulfill its primary function of providing oxygen to the body cells and removing carbon dioxide, a waste product of cellular metabolism. The exchange of gases occurs in two main steps: external respiration and internal respiration. External respiration involves the exchange of gases between the lungs and the blood. It occurs in the pulmonary capillaries which surround the alveoli in the lungs. The process can be summarized as follows, like inhalation. During inhalation, the diaphragm and intercostal muscles contract, expanding the thoracic cavity. This leads to a decrease in intrathoracic pressure, causing air to rush into the lungs. Oxygen-rich air enters the alveoli. Diffusion. Oxygen moves from the alveoli into the capillaries where it binds to hemoglobin in red blood cells. At the same time, carbon dioxide, a waste product, moves from the capillaries into the alveoli. Then oxygen transport. Oxygenated blood is carried away from the lungs by the pulmonary veins and is then pumped by the heart to the body's tissues. Then carbon dioxide removal. As oxygen is delivered to the tissues, carbon dioxide is produced as a waste product. Carbon dioxide diffuses from the tissue into the capillaries where it binds to hemoglobin or is converted into bicarbonate ions for transport back to the lungs. Then internal respiration. Internal respiration refers to the exchange of gases between the blood and the body's tissues. This process occurs in the systemic capillaries where oxygen is delivered to the tissues and carbon dioxide is removed. The steps involved are as follows: oxygen delivery. Oxygen dissociates from hemoglobin in capillaries and diffuses into the tissues where it is used for cellular respiration. Carbon dioxide removal. Carbon dioxide, a byproduct of cellular respiration, diffuses from the tissues into the capillaries. It is then transported back to the lungs to be eliminated during exhalation. Several factors contribute to efficient gas exchange, like surface area. The large surface area of the alveoli provide ample space for gas exchange to occur. Then thin membrane. The alveolar and capillary walls are thin, allowing for the rapid diffusion of gases across the membrane. Then partial pressure gradient. Gases move from areas of higher partial pressure to areas of lower partial pressure, facilitating diffusion across the alveolar capillary membrane. Then hemoglobin binding. Oxygen binding to hemoglobin in red blood cells, increasing the amount of oxygen that can be carried in the bloodstream. Then respiratory rate. The rate and depth of breathing affect the exchange of gases. Increased respiratory rate allows for more efficient gas exchange. Overall, the exchange of gases is a vital process that ensures the delivery of oxygen to the body's tissues and the removal of carbon dioxide. It involves the coordinated efforts of the respiratory system, cardiovascular system, and cellular metabolism to maintain proper oxygenation and gas balance throughout the body. Then, partial pressures involved in lung and tissues. In the context of gas exchange, partial pressure refers to the pressure exerted by a specific gas within a mixture of gases. The partial pressure of oxygen and carbon dioxide play a crucial role in the exchange of gases between the lungs and the tissue. In the lungs, oxygen partial pressure. Alveolar oxygen partial pressure, or PaO2, is the partial pressure of oxygen in the alveoli of the lungs. Under normal conditions, it is approximately 100 mmHg. Oxygen partial pressure in the pulmonary capillaries. It represents the partial pressure of oxygen in the blood as it leaves the lungs and enters the systemic circulation. Its value is around 95 to 100 mmHg. Carbon dioxide partial pressure. Alveolar carbon dioxide partial pressure. This is the partial pressure of carbon dioxide in the alveoli. Normally it is approximately 40 mmHg. Then carbon dioxide partial pressure in the pulmonary capillaries, PaCO2. It represents the partial pressure of carbon dioxide in the blood as it enters the pulmonary capillaries. Its value is around 40 mmHg. In the tissues, oxygen partial pressure in the systemic capillaries, PaO2. As oxygen is delivered to the tissues, its partial pressure decreases. In the systemic capillaries, PaO2 is around 40 mmHg. Carbon dioxide partial pressure. Carbon dioxide partial pressure in the tissues, PvCO2. It represents the partial pressure of carbon dioxide in the tissues. Under normal conditions, it is approximately 45 mmHg. Carbon dioxide partial pressure in the systemic capillaries, PaCO2. As carbon dioxide is produced by cellular metabolism, its partial pressure increases in the systemic capillaries. Its value is around 45 mmHg. The partial pressure gradients between the lungs and the tissues drive the diffusion of gases. Oxygen moves from areas of higher partial pressure in the lungs to areas of lower partial pressure in the tissues, while carbon dioxide moves in the opposite direction. It is important to note that these values are approximate and can vary depending on factors like altitude, exercise, and certain medical conditions. Additionally, partial pressure of gas are measured in millimeters of mercury or kilopascals and are commonly determined using arterial pressure gas analysis. This helps to learn about the efficiency of gas exchange and diagnosing respiratory disorders. Monitoring these values is crucial in medical settings to ensure appropriate oxygenation and ventilation of the body's tissues. Then, oxygen dissociation curve. The oxygen dissociation curve, also known as the oxyhemoglobin dissociation curve, is a graphical representation of the relationship between the partial pressure of oxygen and the percentage of hemoglobin that is bound to oxygen in the blood. It illustrates how the affinity of hemoglobin for oxygen changes with varying levels of oxygen partial pressure. The shape of the oxygen dissociation curve is sigmoidal or S-shaped. At low partial pressures of oxygen, such as in tissues, the curve is relatively flat, indicating that hemoglobin has low affinity for oxygen and does not readily bind it. At the partial pressure of oxygen increases, such as in the lungs, the curve steepens, indicating that hemoglobin's affinity for oxygen increases, resulting in more efficient oxygen uptake. The key factors influencing the shape and position of the oxygen dissociation curve are hemoglobin's affinity for oxygen. Hemoglobin can exist in two states: oxygenated oxyhemoglobin and deoxygenated deoxyhemoglobin. The binding of oxygen to hemoglobin is influenced by factors such as pH, temperature, carbon dioxide partial pressure, and the concentration of 2,3-diphospho-glycerate 2,3-DPG in the red blood cells. These factors collectively affect the affinity of hemoglobin for oxygen. Bohr effect. The Bohr effect describes the influence of pH on the oxygen hemoglobin affinity in tissues with high carbon dioxide levels and low pH. Due to increased metabolic activity, the oxygen dissociation curve shifts to the right, indicating a decreased affinity for hemoglobin for oxygen. This shift promotes the release of oxygen to the tissues, facilitating oxygen unloading. In tissues with high carbon dioxide levels and low pH due to increased metabolic activity, the oxygen dissociation curve shifts to the right, indicating a decreased affinity of hemoglobin for oxygen. This shift promotes the release of oxygen to the tissues, facilitating oxygen unloading. Temperature. An increase in temperature decreases the affinity of hemoglobin for oxygen, causing the oxygen dissociation curve to shift to the right. Increase in temperature decreases the affinity of hemoglobin for oxygen. This allows for increased oxygen release to the tissues during exercise or elevated body temperature. Carbon dioxide partial pressure. An increase in carbon dioxide partial pressure as occurs in tissues decreases hemoglobin's affinity for oxygen, resulting in the rightward shift of the oxygen dissociation curve. This phenomenon is known as the Haldane effect. Then the significance of oxygen dissociation curve lies in its ability to ensure efficient oxygen delivery to the tissues. The shape of curve allows for oxygen uptake in lungs, high affinity, and oxygen release in the tissues, low affinity. This ensures that oxygen is readily unloaded where it is needed most, such as during exercise or in metabolically active tissues. The oxygen dissociation curve also helps in understanding respiratory and cardiovascular disorders. Shifts in the curve, such as a rightward shift due to factors like increased acidity or temperature, can impair oxygen delivery to tissues and indicate compromised respiratory function. So, in summary, The oxygen dissociation curve illustrates the relationship between oxygen partial pressure and the binding and release of oxygen by hemoglobin. It is influenced by factors such as pH, temperature, and carbon dioxide partial pressure. Understanding the curve helps in assessing oxygen transport, tissue oxygenation, and diagnosing respiratory and cardiovascular conditions. Then, factors affecting binding of oxygen to hemoglobin: PO2, PCO2, PCO, pH in detail with detailed explanation. The binding of oxygen to hemoglobin is influenced by several factors, including the partial pressure of oxygen, partial pressure of carbon dioxide, concentration of carbon monoxide, and pH. These factors play a crucial role in regulating the oxygen hemoglobin dissociation curve and ensuring efficient oxygen transport in the body. So, partial pressure of oxygen, PO2. PO2 represents the concentration of oxygen dissolved in the blood. As the PO2 increases, hemoglobin has a higher affinity for oxygen, resulting in more oxygen binding to hemoglobin molecules. This relationship is depicted by the oxygen hemoglobin dissociation curve. In the lungs, where PO2 is high, hemoglobin binds to oxygen readily, facilitating oxygen uptake. In the tissues where PO2 is higher, PO2 is lower, hemoglobin releases oxygen to meet cellular demands. Next factor, partial pressure of carbon dioxide or PCO2. PCO2 represents the concentration of carbon dioxide dissolved in the blood. Increased PCO2 causes a decrease in blood pH, leading to a phenomenon known as Bohr effect. The Bohr effect reduces the affinity of hemoglobin for oxygen, causing a rightward shift of the oxygen hemoglobin dissociation curve. This shift promotes oxygen unloading in tissues with high levels of carbon dioxide and increased metabolic activity. Then, concentration of carbon monoxide. Carbon monoxide is a toxic gas that binds strongly to hemoglobin, forming carboxyhemoglobin. CO has a higher affinity for hemoglobin than oxygen, so even at low concentrations, it can significantly reduce the oxygen carrying capacity of the blood. This can lead to tissue hypoxia and severe health consequences. Carbon monoxide poisoning is a medical emergency that requires immediate treatment with oxygen to displace CO from hemoglobin. Then pH. The pH of blood is influenced by the concentration of carbon dioxide and bicarbonate ions. Acidic conditions, which create low pH, promote oxygen unloading. Oxygen unloading. While alkaline conditions, high pH, enhance oxygen binding to hemoglobin. This relationship is important in maintaining the balance between oxygen uptake in the lungs and oxygen release in the tissues. Acidic conditions in metabolically active tissues facilitate oxygen delivery, ensuring that oxygen is readily available for cellular respiration. Then the importance of these factors lies in their role in regulating oxygen transport and tissue oxygenation. The interactions between PO2, PCO2, CO, and pH help to optimize oxygen delivery to tissues. For example, during exercise, increased carbon dioxide production and acidic conditions in muscles promote oxygen release, ensuring an adequate oxygen supply for energy production. In contrast, in the lungs, where PO2 is high and PCO2 is low, oxygen binds to hemoglobin, enabling efficient oxygen uptake. Understanding these factors is crucial in diagnosing and managing respiratory and cardiovascular disorders. Alterations in any of these factors can disrupt the oxygen hemoglobin equilibrium, leading to impaired oxygen transport and tissue hypoxia. Monitoring blood gases, including PO2, PCO2, pH, is essential in assessing respiratory function and acid-base balance. Then factors affecting binding of oxygen to hemoglobin: body temperature, diphosphoglyceric acid levels, total hemoglobin, and also myoglobin. In addition to the factors mentioned earlier, PO2, PCO2, CO, and pH, several other factors influence the binding of oxygen to hemoglobin. This include body temperature levels of 2,3 diphosphoglyceric acid or DPG, total hemoglobin concentration and the presence of myoglobin. Understanding the role of these factors is important as they contribute to the regulation of oxygen transport and tissue oxygenation. Body temperature. Body temperature affects the binding of oxygen to hemoglobin. As temperature increases, the affinity of hemoglobin for oxygen decreases. This phenomenon is known as Bohr effect. A higher body temperature shifts the oxygen hemoglobin dissociation curve to the right, promoting oxygen unloading in tissues with increased metabolic activity. This ensures that oxygen is readily available for cellular respiration and oxygen production. Then 2,3-diphosphoglyceric acid or DPG. DPG is a byproduct of red blood cell metabolism. It plays a crucial role in regulating the affinity of hemoglobin for oxygen. Increased DPG levels decrease the affinity of hemoglobin for oxygen, promoting oxygen release to tissues. DPG levels are influenced by factors such as altitude, chronic lung diseases, and anemia. For example, in individuals living at high altitudes where oxygen availability is reduced, the body compensates by increasing DPG levels, enhancing oxygen unloading and improving tissue oxygenation. Then total hemoglobin concentration. The total concentration of hemoglobin in the blood affects its oxygen carrying capacity. An increase in total hemoglobin concentration can enhance the oxygen carrying capacity, enabling more oxygen to be transported to tissues. Conversely, conditions that result in reduced hemoglobin levels, such as anemia, can impair oxygen delivery to tissues and lead to tissue hypoxia. Then myoglobin. Myoglobin is an oxygen binding protein found in muscle tissues. It has a higher affinity for oxygen than hemoglobin and acts as a reserve for oxygen storage in muscle cells. During periods of increased oxygen demand, such as exercise, myoglobin releases oxygen to meet the metabolic needs of the muscles. This allows for efficient oxygen delivery within the muscle tissue and helps to sustain aerobic respiration. The importance of these factors lies in their contribution to the regulation of oxygen transport and tissue oxygenation. By modulating the affinity of hemoglobin for oxygen, these factors ensure that oxygen is readily delivered to tissues with varying metabolic demands. They help optimize oxygen uptake in the lungs and oxygen release in the peripheral tissues, ensuring efficient oxygenation and cellular respiration. These are very important, like for instance in the field of sports medicine. Knowledge of the influence of body temperature, DPG levels, and myoglobin on oxygen transport helps athletes optimize their performance. Altitude climbers and individuals with respiratory or cardiovascular conditions can benefit from understanding the impact of these factors on oxygenation and adapting their activities accordingly. Furthermore, monitoring total hemoglobin concentration is important in the diagnosis and management of anemia, which can significantly affect oxygen delivery to tissues. So in conclusion, the binding of oxygen to hemoglobin is influenced not only by PO2, PCO2, CO and pH, but also by factors such as body temperature, DPG levels, total hemoglobin concentration, and the presence of myoglobin. These factors contribute to the regulation of oxygen transport and tissue regional oxygenation, ensuring efficient oxygen delivery to meet the metabolic demands of the body. Understanding the role of these factors is essential in various fields, including medicine, sports, and altitude-related activities, as they help optimize oxygenation and improve overall health and performance. Next, neural regulation of respiration in detail. Neural regulation of respiration is a complex process that involves the coordination of various neural pathways and centers in the brain to control the rate and depth of breathing. The primary centers responsible for the regulation of respiration are located in the medulla oblongata and the pons of the brainstem. The centers receive inputs from various sensory receptors and generate outputs that stimulate the muscles involved in respiration. First, medullary respiratory centers. Dorsal respiratory group or DRG. Dorsal respiratory group. It is located in the dorsal part of the medulla and is responsible for the generation of basic rhythmic pattern of breathing. It sends impulses to the inspiratory muscles, primarily the diaphragm, to initiate inspiration. Then ventral respiratory group or VRG. So dorsal respiratory group is located in the dorsal part of the medulla and is responsible for the generation of the basic rhythmic pattern of breathing. It sends impulses to the inspiratory muscles, primarily to diaphragm, to initiate inspiration. Then ventral respiratory group VRG is located in the ventral part of the medulla and plays a role in both inspiration and expiration. It provides additional respiratory drive during deep breathing and activates the expiratory muscles during forced expiration. Then pneumotaxic and apneustic centers. Pneumotaxic center. The pneumotaxic center is located in the upper pons and helps regulate the duration and intensity of each breath. It acts to inhibit inspiration, limiting the depth of breathing and promoting faster respiratory rates. Then apneustic center. The apneustic center is located in the lower pons and has an excitatory effect on inspiration. It stimulates the inspiratory centers in the medulla, prolonging the inspiratory phase of respiration. Then chemoreceptors. Central chemoreceptors. Central chemoreceptors are located in the medulla and respond primarily to change in the pH of the cerebrospinal fluid. An increase in carbon dioxide CO2 levels in the blood leads to increased carbonic acid formation, resulting in a decrease in CSF pH. This stimulates the central chemoreceptors, which in turn increase the rate and depth of breathing to eliminate excess CO2 and to restore normal pH levels. Then peripheral chemoreceptors. Peripheral chemoreceptors are located in the carotid bodies and aortic bodies. They are sensitive to changes in the partial pressure of oxygen and carbon dioxide and pH in the arterial blood. When arterial PO2 decreases or arterial PCO2 or pH increases, the peripheral chemoreceptors send signals to the medullary respiratory centers, resulting in an increase in respiratory rate and depth. Then stretch receptors. Pulmonary stretch receptors. Pulmonary stretch receptors are located in the smooth muscles of the airways and respond to the stretch of lung tissues during inflation. When these receptors are stimulated, they send inhibitory signals to the medullary respiratory centers, resulting in a decrease in inspiratory activity and preventing overinflation of the lungs. Then Brewer reflex. Brewer reflex. The Brewer reflex is a protective mechanism that prevents overinflation of the lungs. It is mediated by the activation of stretch receptors in the lung tissue, which sends inhibitory signals to the medullary respiratory centers. This reflex is particularly important in controlling respiratory rate and depth during activities such as exercise. The neural regulation of respiration is essential for maintaining adequate oxygenation and carbon dioxide elimination of the body. It ensures that the respiratory rate and depth are appropriately adjusted to meet the metabolic demands of the body. Any disruption in this regulation can lead to respiratory disorders such as hypoventilation or hyperventilation. So in summary, the neural regulation of respiration involves the coordination of medullary and pontine respiratory centers as well as various sensory receptors, including chemoreceptors. Next topic, chemical regulation of respiration. Chemical regulation of respiration plays a crucial role in maintaining the appropriate levels of oxygen and carbon dioxide in the body. It involves the detection of changes in blood gas concentrations by specialized chemoreceptors and the subsequent adjustments in respiratory activity to restore homeostasis. Location. The central chemoreceptors are located in the medulla oblongata. More details about the chemical regulation of respiration will be dealt with the help of next video. Thank you for listening.